Hello, welcome to another video from the conscientious biologist, Ben Gallagher. This one's really interesting. It's one of my favourite bits of biology because its potential is huge. This is about monoclonal antibodies. These are something that can be created and used to cure loads and loads of communicable diseases or to relieve the symptoms of them by dealing with the toxins. This is part of the GCSE syllabus. It also crops up at A level though, and the theory really is about the same. As always, I'll ask that you please do subscribe to my channel on YouTube if you haven't done so already. That will keep you updated on videos as I make them. Thank you. So before we move on to monoclonal antibodies, let's just refresh our memories on what antibodies are and what they do. They're vital for the immune system. So the first key point about antibodies is that they're proteins. I've told you so many times throughout this course that proteins do everything. Really, your cells in your body is totally controlled and functions because of the proteins that you can make using the instructions from your DNA. Now, like all proteins, it's their shape that determines their function, and their shape is roughly like this above me. There's a long kind of thin bit, and then there's a head that can be of variable shapes. So that top bit is the variable part, the bottom bit is the constant part, and that top bit, the variable part, can be complementary in shape to an antigen on a specific pathogen. And remember, antigens are just things on the surface. So what we're talking about, it's complementary to the body of the pathogen. So there'll be a part of the pathogen that sticks out that variable group can attach onto. Think again what I just said there, they'll attach to the body of the pathogen, hence antibody. But some variable groups on some antibodies can attach to the pathogen's toxins. So like you're seeing here, pathogens can release poisons and toxins, these little red stars that I've put on there, that kind of poison your body, and that's what can make you really ill, and in some cases that's what can kill you. Now, because some antibodies can attach to the toxin, that's where you call them antitoxins. So notice I've changed it now, it's antibodies and antitoxins, same basic molecule, but if they bind to the body of the pathogen, they're an antibody. If they bind to its toxin, they're an antitoxin. It's pretty straightforward stuff. The constant part of the antibody is always the same. No matter which lymphocyte made it, the shape of the constant part is always the same in each organism's body. So your constant region might be different to mine, but every single antibody in your body will have the same constant region. Now let's look at why that's important. So different lymphocytes within your body can make different antibodies, but they always make the same ones. So these two lymphocytes that I've put over there, you can see the first one's got kind of a bowl-shaped variable part, the next one's got more of a spiky part, but each one only makes its own type of antibody. Now, if a pathogen comes along and it encounters that first lymphocyte, it's the wrong shape to fit that antibody. But if that pathogen encounters the second one, it activates it, which causes that lymphocyte to rapidly multiply by mitosis until you've got loads and loads of clones of that original lymphocyte, all making exactly the same antibody and releasing that antibody into the blood. That's really important because that antibody is specific to the pathogen that first stimulated the lymphocyte, which is the pathogen that's now inside your body that needs dealing with. Now, why those antibodies are so important is because they will bind to the pathogens that are inside your body. Now, remember the constant region we talked about before. So I've made it flash over there. Look, that constant region can be easily recognized by a phagocyte. The phagocyte can then come along and do phagocytosis, which you saw in disease 5, the white blood cells video, swallow those pathogens really quickly and deal with the pathogenic infection very rapidly and make you better. Okay, now remember the pathogens might also release toxins. The lymphocyte I've got up the top there, look, its antibodies are specific to the toxins, so that the toxin binding to that lymphocyte would activate that lymphocyte. It would then put antibodies into the blood as well. They'd bind to the toxins and the phagocyte can deal with those toxins as well. The key thing then about lymphocytes is that flooding the blood with antibodies for the specific pathogen is the most effective way to fight infection. If you can get loads and loads of the antibodies that are specific to that pathogen into the blood, that bind to the pathogen, bind to its toxins, massively speed up the response of the phagocytes, and you'll get better very quickly. So flooding your blood with antibodies, big bonus for getting better. So let's now talk about monoclonal antibodies. Let's first just explain that word. Mono, you've probably come across before. Mono means one. Clonal, or clones, means lots of copies of the same thing. So monoclonal basically means lots of copies of one type. Okay, exactly what you see in a lymphocyte response when a lymphocyte's been stimulated and it multiplies and it releases all those identical copies of antibodies into your blood. So monoclonal antibodies are just loads of copies of one type. It's exactly what you see in a lymphocyte response. But these are things that we can make artificially. So what you need to start this off, you need an animal with roughly a similar immune system to us as humans. So most mammals will do for this. I've picked a little rabbit, no particular reason at all, but it serves its purpose. So what we need to do first, we need to give that animal the pathogen that we need immunity from. So you inject that animal either with the pathogen itself to make the animal ill, or a vaccine. Hopefully you've already watched the vaccines lesson, it was the one before this. Either way, the pathogen itself or the vaccine for that pathogen should stimulate an immune response in that animal, which means it will activate a lymphocyte. The lymphocyte will multiply by mitosis. It will create the antibodies, and those antibodies will go and flood the blood. Now, those antibodies are making, those lymphocytes are making the antibody for that pathogen. So if you can extract from that animal's blood one of those lymphocytes that has the specific antibody, then you've got the antibody that you need to treat that infection. But of course, one lymphocyte alone isn't going to do you any good, because the lymphocytes formed after the rapid mitosis, after activation of the lymphocytes, all those loads and loads of copies that you can see over there, their job is to rapidly make loads and loads of antibodies, but then they die. Okay, so that cell that we've extracted can't multiply now on its own. It won't naturally do mitosis and copy itself. The original one that triggered the response would, but all the clones that get made from the original, they lose the ability to do mitosis themselves. So step 4, you need to get a tumor cell. So tumor, like we're talking about cancer cells, okay? Tumor cells can do mitosis very rapidly. So what you do, you fuse this tumor cell to your lymphocyte to create this cell. This is called a hybridoma because it's a hybrid between a tumor cell and the lymphocyte. Those cells can multiply very rapidly, and in multiplying, they will then start to produce loads and loads of copies of the antibody. And remember, those antibodies, it's lots of one specific type. In other words, monoclonal. So all those antibodies are going to be produced by all these copies. Now, this isn't done inside an organism before anyone panics and goes, what, but that's tumor cells, that's cancer cells. Since we removed the cell from that rabbit, everything else is done in the lab, in a petri dish, under controlled conditions. So you're talking about these cells above my head, all multiplying and producing their antibodies in containers, not inside living things. So there's no risk of actually giving something a cancer now. Plus, you don't even use these cells. All you need is the antibodies. So all you need to do as the final step is extract all the antibodies, purify them, and bottle them and package them up. So what I've got in this vial beside me now is a liquid that would just contain billions of the specific antibodies for the pathogen that we originally gave to the rabbit over there. That's what monoclonal antibodies are. Loads and loads of copies of one type of antibody specific to one pathogen. Take a screenshot of this because this is the really important bit from this presentation. Right, hopefully you've already kind of gone, oh, that's really useful because that means we can use them for this reason. But let's just make it really obvious and let's run through the uses of monoclonal antibodies. There's two main uses. The first one is in fighting disease. So let's take this person here. They're looking very happy, so let's ruin their day and let's give them a pathogenic infection. So if that person catches a communicable disease by whatever reason, they breathe it in or from direct contact or it's in their food or whatever reason, they've contracted a pathogenic disease. That pathogen has multiplied inside them, it's now in them. That pathogen could multiply to a level where it could make this person really ill, or it could kill them. It could be a life-threatening thing. Now, hopefully, that person will have a natural immune response. Their lymphocytes will be triggered. Their own lymphocytes will release loads of antibodies, bind onto all those pathogens, bind onto their toxins, and their own phagocytes will deal with it. But that doesn't always happen. Sometimes the phagocytes, the pathogens, multiply too rapidly, and they just swamp the person's immune system, and you lose that war, and the person could lose their life. This is where monoclonal antibodies come in. If we've got the antibodies specific for that pathogen that the person's got, which of course we could get by taking that same pathogen, giving it to the animal, to the rabbit, using that process we've just seen, we can get them like this. Now, if those are injected directly into that person, then those antibodies in the person's blood will tag onto the pathogens in exactly the same way as if this person had made their own antibodies. You basically mimic the natural process. We said on the first slide, flooding the blood with antibodies is the best way to fight off infection. That would happen naturally, but if it doesn't, you can just flood the blood artificially by injecting them directly into you. Now, if they've tagged all the pathogens, of course, the phagocytes can then come along really quickly and easily, munch all the pathogens, and the person's cured, the person's better. So monoclonal antibodies, really, really quick way to fix and cure someone of a disease. And this can work for almost any communicable disease, be it viral, fungal, or bacterial. If we can make monoclonal antibodies for that pathogen, then that person becomes ill with that pathogenic disease, inject them with the specific monoclonal antibodies to help their immune system recognize it and fight it, and they should get better quite quickly. Now, of course, the problem with this is that the person that we've injected hasn't actually developed immunity of their own. Because they didn't have their own lymphocyte response, they won't have had a chance to develop any memory cells. So if that same pathogen infects them again, they're still at every single bit as much of a risk as the first time they've got it. Of course, you could just treat them with monoclonal antibodies again. But to be honest, if it's a disease that's very life-threatening, something like Ebola, if Ebola get into you, it will kill you before you're likely to have a lymphocyte response. If we've got monoclonal antibodies for it, inject the person with monoclonal antibodies, they'll get better, they'll survive, then hopefully they can move away from where the danger area was and not catch Ebola again. Okay, but the key thing with this, they don't develop their own resistance. Okay, really quickly, second is as not monoclonal antibodies, but as monoclonal antitoxins. Now, they're the same thing. They can be got in the same way, but these can work for if someone's got toxins in their body that need to be sorted out. And they don't have to be pathogenic toxins. So again, same person here, they're again looking happy. Let's ruin their day again. Let's say they've just been bitten by a highly venomous rattlesnake or maybe a black widow spider. That means the toxins from those will be in their blood. Just like bacterial toxins can be in their blood, these could be toxins from other creatures, because almost all toxins and venoms are protein based. These creatures will have made them because they will have genes in their DNA that code for the protein that is the toxin. The ribosomes make the toxin, make the protein, and they inject it into the person. Okay, and like all proteins, it's their shape that determines their function. And if the toxins have a shape, that shape can be matched and be complemented by an antibody. So now, if I've got a little container, a little bottle of complementary antibodies, you'll notice in this bottle they're different shaped antibodies. They're the shaped ones for the toxins that we were looking at on the first screen. But the same process, if we inject this person with those monoclonal antibodies, they will bind to the toxins. They can directly neutralize the toxin and stop it working, or they can attract phagocytes and the phagocytes will deal with the toxin. But either way, that person's now not going to be affected by those toxins. You've probably heard of this in films and things like that, where you hear about things like an antidote, or quick, get the anti-venom or something, and people get bitten by spiders in the wild and they go, oh, quick, give them an injection of anti-venom. This is what we're talking about. We're talking about monoclonal antitoxins that have been specifically made for the specific snake or the specific spider or the specific whatever poisonous venomous creature that it was. Okay, hopefully that's quite straightforward, and that's it really for monoclonal antibodies. You can use them against disease, you can use them against toxins. They're absolutely brilliant tools in the fight against disease and infection. Now that leads us really nicely into one of the final videos on disease. So this one looked at how monoclonal antibodies can be used as a type of medicine. In the next video, we look more specifically at more traditional medicines and drugs that we think of as the things that you take to make you better when you're ill. But we're focusing in that one on the drug development system. How does a drug get discovered and someone think, well, this might work as a drug, right through to the stages where it's commercially available to everyone to use. That's again from the GCSE syllabus, and it just continues on your disease unit. If you haven't done so already, as always, please do subscribe to my YouTube channel. Give this video a like if you found it useful. Share it with friends. It's really interesting and really good common knowledge stuff that people should have. Also, head over to my Facebook page where there's loads of infographics and extra bits of biology to round out your learning. Thank you.